It is, from the point of view I've made up, I have no reference because you don't have a reference for something like this. It's my knowledge of scale of hands to baby, you know, my knowledge of female anatomy. I know it has to do with the fetal spine and the brachial plexus, and this is the muscle that gets affected and paralyzed and for injury. I have gotten down into subcellular and some molecular. This is a genetically engineered cancer drug. Don't do it very often, but can do it. Look at all those conventions I've used up there. You know, I'm not dealing with molecular data. All right, so this is an example of an illustration or a drawing that we as an illustrator we use to communicate to an animator. It's very often when people are doing, when there's medical animations being made, that the animators have no medical knowledge whatsoever. They may have studied something completely different. So a medical illustrator is vital in showing them and telling them about the science and about the aesthetic qualities of, you know, and storytelling of what's going on. So this is actually for Tropicana, and it was all about calcium being absorbed. Being absorbed in the intestinal lining. So while this is not a traditional storyboard, meaning that it's not telling the animator, shoot it from this way, shoot it from this way, here's the script as it's going along. It's informing this really super skilled, fantastic animator that we're working with, what the anatomy is, what he needs to be considering, what the structures are, how this passes through the outer villi to the cellular level, and how it gets incorporated into the cell. And here is this. I don't think this is the final video. It has no music. It has no narration, but you can see what he did with the information that we provided.
Is that what the inside the intestine?
Yes. All those villi increase the surface area for absorption. So those are calcium.
Oh.
Coming in. And you notice how they avoided the whole cellular thing. Like they came down, you know, and they did like a really interesting point of view, but it's kind of lacking in the content. Yeah, but that's, you know, that's what happens. All right, we'll move on to imaging. All the changes that have happened in biovisualization are occurring because of this idea of imaging. As that is evolving, our understanding of structures are evolving. CT, everyone's probably familiar with that. PET, MRI. MRA is same thing as an MRI, but it's with an angiogram, meaning that contrast is injected into your brain. This SEM is scanning electron microscopy and X-ray crystallography. Scanning electron microscopy has been around for a while, but this X-ray crystallography is really what has driven all the understanding of biological molecules. And it's this, don't ask me about the science behind it, it's extremely complicated, but what it basically does is that they grow these things in crystals and then shoot electrons at it, and it gives them the structures of molecules. Say that again. Yeah. They grow like molecules, I'm going to use that as a term, in crystals. So they don't always know what's going to form in these crystals, and they shoot electrons at it, or X-rays at it, and it gives them a visual picture of atomic relationships in molecules. I know, whoever you know who that is. Then there's also this idea of software that has been developed that allows us to access this research and access these images and access these structures. All right, light microscopy, you're all familiar with that. So in terms of structure, we're starting to understand all the way down to the molecular level. And as our understanding of structure has grown, our understanding of function grew too, because we understood how proteins interacted with each other, how they fit into each other, and it was very eye-opening. This is actually a scanning electron microscope picture. This is an algae, like a coccolith, I believe it's called. And the reason why I put it up there is purely because there are really unbelievable images of all kinds of nature in scanning electron microscopy. They're very cool. All right, this is just the light microscopy. This is what I would have used in determining cell types and stuff. One of many images that would have helped me in doing that cervical cancer piece. So I'm interested in, amount of cytoplasm, a nucleus, outer cell. Notice that halo right there? That's HPV. Cells with HPV create this halo around the nucleus.
The human papillomavirus?
Yep. Which is a cause of dysplasia and cervix. All right, here's some more of those coccoliths. Aren't they just so cool?
Yeah.
So it's kind of arbitrary and throwing them in here, but they're just spectacular. And there's tons of these images.
It looks like, fun cakes. What are they? Are these
That's algae. Like it's like a cytoskeleton of an algae. Blood. So when they were talking about the leukocytes in the inner light, this is a leukocyte. And notice its scale to a red blood cell. So imagine your blood streaming through, that's just packed with blood cells and plasma, platelets. There's platelets on here. Plasma's the liquid that are all streaming through the little capillary, and all they eliminated to show you in that movie to just see the leukocyte against the wall. All right, this is how I learned about cell biology, which is a scanning electron micrograph of a cell. This is a bilobar leukocyte, and here's the whole cell. It's jam-packed, okay? So everything that he was showing in that video, here's those vesicles, those ones that are going to pull along by the microtubules, like your blood cell. They're all these little black, dark spots down here. These up here that look like they have little striation in them, those are mitochondria, ones that are changing shape and going through. But look at all this cellular matrix here. This is all those tubules and that, everything that's going on in there. So this is how I learned about the cells, which is very different than watching a really cool animation. All right, so this is an illustration of mine that is early, but notice how it has it has a nucleus, it has the endoplasmic reticulum, it has the Golgi, it has the mitochondria, it has the vesicles. And you notice this cytoplasm has this sense of like the sack of water kind of thing. And that was what everyone's understanding. We knew there was stuff in there, and we knew that there was structure in there, but everyone kind of more or less depicted this, sack of water. And he's in David Linsky's video. He's trying to explain all that along with function. All right, so here's like a shot from the movie about all that structure. Now, I think this is a great shot showing you how much he had to eliminate to give you a sense of all like that fibrous network. All right, so everything that's driving our understanding is taking us to a new level in the cellular stuff is understanding proteins and the cellular structure of proteins and the molecular structure of proteins. So suddenly we can tell what they actually look like. instead of what our artistic interpretation of what a molecule looked like. And this is really driving everything. All right, so in David Blinsky's quote, he refers to a protein dating and accessibility to people of this database. There is this protein data bank that is free, online. Anyone can access it. They have 68 or almost 69,000 molecules up there right now. It's the primary repository of atomic structures of biologically important molecules. Using it is interesting. You kind of have to know what you're doing, and I can't say that I feel completely knowledgeable about this stuff, but you can log in and you can search for different things. I'm going to search for an antigen, immunoglobulin A. And here it gives me the entries. So I'm going to narrow it to human. Okay, so if you'll notice all these things come up. The trouble, all these are based off X-ray, crystallographic images. That's how they get this information. Now, you notice they all look different. Hello, what does that mean? So many of these things are just functional segments and subunits of molecules. This is the danger in just using it and applying it. You can easily make a giant horrific mistake about a molecule that is really only a part. So it needs to be supplemented with other information. So it can tell me that this antibody is important in kidney disease. I can search through other information about it, but here is what it looks like. Now I can view this interactively. Right, here's what it looks like. I can get a sense of it in space. You know, I'm so not good at this moving things, based off this 3D, but we can look at it differently. Those of you who have had any chemistry, we can look at things in the ball. These colors relate to different functional chains within. We can look just at the backbone of the chains. You can also export this information into 3D programs, which is why it's become so valuable. Now imagine every molecule that was done in inner life started with something like this. And they incorporated every coordinate for every molecule, every atom, and that's how they built the movie. So, this is really pretty spectacular to have this access. I'm going to do one more. And just another antibody. This one's just got a cool crystal 1,999 atoms. So things that are larger than that are only the fragments. Larger structures aren't shown. Some things like antibodies have large flexible sections that bend and twist, so often they'll only show, they're hard to crystallize, you know, so they'll only show like functional units. But you know, you always need to refer back to other things and don't take it as, you know, informing you completely. All right, so our understanding of those molecular structures has really, really increased our knowledge of function. And the ability to go right to 3D allows us to do simulations of proteins where they can take 3D models of one thing and another thing and see how they interact and link together. And you saw evidence of that in real life. Here's just another shot of what, so this is a protein that's embedded in the cell membrane. So now that we understand the physical structure, we can think about how things interlock and open up channels and change our understanding. This is just another shot showing you, oh, look at them all. Like, there's a ton of molecules in there. Yeah. And they, you know, granted they're the same ones, probably repeated. They may have slightly different functions. We see some other embedded kind of proteins in here, but making the decisions about what to eliminate and what to keep is just mind-boggling. All right, so our understanding, Drew has taken all this to a new level. He just won the MacArthur Genius Award.
Oh my God,
okay? He is a medical animator. He works at the Walter and Eliza Hall Institute of Medical Research. And I'm going to show you one of his awesome videos. Hang on a second, I'm going to try to write it up because you will not appreciate it. I already don't know how to do that.
What are you trying to do?
Right.
Oh, Bob knows.
Right next to me.
This is why you take our history and transition.
Great. And then I'll need it.
Mosquitoes are usually vegetarian, preferring to drink nectar, fruit juices, and honeydew. Only a pregnant mosquito will bite humans, seeking nutrients from blood to nourish her developing eggs. If she drinks blood from someone infected with malaria, she too becomes infected with the disease. The tiny drop of blood filling the insect's stomach is teeming with malaria parasites. The parasite form that is deadly inside humans cannot survive in a mosquito's stomach and is slowly digested with the rest of her blood meal. However, back in the human host, a few of the parasites turn into a different type of cell, one that is sexual but remains dormant. Malaria sex is triggered when the warm human blood begins to cool inside the insect's stomach. The female form of the parasite matures into an egg. The male form takes a while longer to mature into sperm. This sperm is from an earlier feed. The fertilized cell can glide and begins to explore its new environment. It migrates to the outer lining of the mosquito's stomach before transforming into a cyst. Each cyst produces thousands of thin, tiny parasites which seek out and infest the mosquito's salivary glands. The next time this mosquito bites a victim, the malaria parasite will ride in with her saliva and infect another human. This year, 10% of people on Earth will be struck down with malaria. Most people who die of the disease will be pregnant women and children under the age of 5.
His work is different in the way that it's the most scientifically accurate. You could be the most up-to-date information about molecular shape, scale, behavior, and spatiotemporal dynamics. So he's adding behavior and studies of behavior along with it. Like the movement of that mosquito was based off actual imaging. So he's adding everything together. That's just one video. He has many, many others, and you can access them online. Because most of the people who do that are working for institutions that are either publicly funded or convention. We talked a little bit about convention. I'm just going to kind of fly through that. Just because you can do something that's molecular imaging, should you do it? And what is, like the average Joe is going to not get anything from that. And how do we make things more accessible to everyday people and understanding? Now, this is actually from Science Magazine, which is not a dummy magazine by any stretch. But notice, proteins are more schematized. And that's just fine. It tells the story of their distribution, what they are. You need labels to distinguish what they're for, but you don't always need the level of complexity that's possible. This is one of Todd Buck's illustrations on rheumatoid arthritis. And when I asked him for an image, I asked him for this one specifically because it tells a story within a single static image. And believe it or not, there is some preliminary research that people actually learn better from this than animation. That when you are viewing things in motion, there's a passiveness to your brain.
I was highly entertained by the
Yes, but when you have to read text and follow through, it awakens other parts of your brain. You're using more parts of your brain. You might actually have better understanding. But no one really has studied this very well, and people are only beginning to really look into it.
I think it's different kinds of information too.
It is.
The video was so much like an experience, and it was like going down the chute.
But the video was made for them trying to think of a better way to teach cell biology to PhD students. So they already have a great understanding and level of understanding, and that has to be taken into account in the audience. This is a very schematized version of antigens. So this is the IgA we looked at, and this is the IgM we looked at.
Now, it's much sexier video.
Yeah, this is what we looked at, the protein database. And this is just fine for most users and most researchers. You know, you don't really need all that. It's really depending on the level of understanding. Do you even need motion to tell a story at all? You know, here is a protein embedded in a layer, much like we saw in the animation, and how this is a sodium potassium pump in and out of the cell. So it's active transport and tells the story through very simple illustrations and may be more effective in learning it. We don't know. People really haven't studied that very well.
You would think it in stages too. I mean, I think it is the intro level, where you want to put everything down, and then the other is so beautiful and showing.
Right. So they're very different, but there's not a good understanding about what's better or what's necessary, or certainly it's very expensive.
That one that we were just looking at, that doesn't make me feel like I made like I have a shimmering body of light.
True.
And the video made me think that I occupy a very peculiar, magical.
And that was part of what their goal was.
Yeah.
To ramp up the mystery of the body and to really get people interested in molecular biology.
Yeah.
Because this really wouldn't make you all jazzed about where you're going.
No. It's not a lot of information. It looks like we're changing the
All right, so this is an image I was showing you earlier. The reason I'm showing you is because there's also this thing called Osiris, which is free as well. You can download the application on the web. What it does is it takes CT, MRI, MRA, ultrasound, angiograms, and allows you to manipulate them. Now the rub is that this information is patient HIPAA protected, and it's an individual we're talking about, an individual patient. But there are some data sets that are available to the public. This may end up being quirky because see, we're starting to scroll through. This is coronal frontal section. This is coronal. I'm just going to get to the meat of this. Okay. So if I can look at this information in different ways, I can play with, as a radiologist would be. This is the same software that the radiologist would be using, by the way. He would be doing it on a more sanctioned machine that is housing patient material, but it's basically the same thing. What is interesting is that this is really changing how I work now. The other was changing how, animators were working, but this is changing how I'm working. What is interesting about it is that I can start to visualize things that are difficult for me to visualize. So that brain that I did before, I was all interested in these vessels in a foreshortened way, and I would have to look at old-fashioned anatomy books and try to envision it, and then my plate cleaners trying to think of them going in space, and now I can play with this. So this is playing with the thickness of the slice. I can play with things like value, trying to get something a little more subtle. Okay. I can look at this in a 3D way. I can rotate it. CT. But you notice right here, that's an aneurysm.
Oh! Burst or waiting? Burst.
No, it's not burst, otherwise it wouldn't see it. Okay, so this allows me, notice I can rotate this, and I can look at it in 3D, and I can do volume renderings on it. And I can export this as an image. I can export it as a PDD. Thing that I can bring into, if I were an animator, I could do such a thing. I could print it out on a 3D printer. So it has given you the ability to make very patient-specific 3D things. So how do I use this? I take it and all right, so here's that image that I was working on. Okay. And I can, so this is my old image. I'm kind of messing with it now and playing with color. I'm starting to repaint stuff because I don't like where things are, the way they looked before. I throw my image that I took from my 3D program, visualization, and I don't put it in there to use it as it is, but I'm starting to paint based off its actual placement rather than my imagined one. But if you look, I wasn't that far off from using my old-fashioned as my visualizing tool. What was different, if you notice, this is the basilar artery that runs right along the brainstem. And if you notice in this, it's shoved way back and up. And I was wrong about that. Like I was a little too bold about its placement. So that's why I'm starting to repaint the brainstem. It's not done, obviously, and I'm trying to shove it back up into the brain a little bit based off what I know. And if you notice, if I reduce this, it's now fitting where my brainstem is.
Oh my God.
So it's really changing the way I'm starting to work. Now, it's very hard to get patient data. I would have to be working with a researcher because of the laws. But I can ask for things, and I can use free databases. It really is only good for imaging arteries, bones, things like nerves cannot be imaged. Veins cannot be imaged because the contrast medium doesn't make it through all the tissue to get to your veins. So there are some limitations.
Can you just dump it in a vein site?
Well, then it would have to go retrograde. Like it would have to
Oh, that's what that was. Swim up there.
All right, so how does this all just transect with art, and how do you guys think about it, and how are other people thinking about it? The Broad Institute, Harvard and MIT, is a whole, it's basically ARTlab in sciences, where it is all about rethinking science and how to learn science and how they are researching in these pods and groups with leaders, and they're taking people from all over with different backgrounds, and they're working together. But they understood this idea that people need to understand the abstract quality of what they're working with. So they have hired medical illustrators to work in their trying to make accessible screens on the outside of their building that are showing exactly what's going on in real time in their research labs. So they work primarily on, they're down to that atomic it's playing live in their blood fever.
What did you say?
As their machines upstairs are churning out the sequence of the genome of the African elephant, they are displaying it live in time in their lobby. And they're understanding here, I'll show you, like it's these. There it is, there. Okay? So they're understanding that people need to have some sort of connection to this abstraction of science. Ben Fry worked at the Broad Institute, and he is a well-known data-driven artist that is visualizing. It's really hard to all this stuff is interactive. It's very interesting. It's all dealing with data and visualization of data. Here's an illustration he did where he's contrasting human DNA with 12 different species, and the farther they, as their sequence changes, it starts to change color, and it represents the distance in evolution to humans. Here's my last attempt at thinking about art and science in a more abstract kind of way, a more conceptual way, just for fun, for our faculty show last year, where I decided to take the elements of blood. There's more cells than that, but some of them are so rare, there really was no point. And take the proportions of them and deconstruct blood and show it in terms of its real cells, but in terms of the data, of the proportions of blood. So just a new way to look at information that's going on.
What are those blue ones down there?
These are leukocytes. This is what they were talking about in life. They are, obviously most of your blood is plasma, the liquid, and red blood cells. And only about 5% of your blood are these leukocytes. Oh, here are platelets. That's what helps your blood clot. So everything is in its proper proportion.
I think that would be a very nice summer shirt. We should actually do one.
I think that's it.
Oh, I was just going to actually ask you about that. That is the strangest thing. I can't imagine how anybody got the idea that you could do that.
Yeah, there's, you think about the people who are, the kinds of people who are doing that are so cerebral, they have no visual sense necessarily.
They don't really have a body.
So, yes, Tim.
You make illustrations, right?
Do what?
You do these kind of illustrations, right?
I do, but I don't work on the molecular level. I mean, once in a while I'll dabble down there, but it's not my comfort zone.
So, if you had to compare yourself, say, some of those like data, and you would say you work more specifically in the nervous anatomy, or like?
No, I work in all over the place. That's part of what the profession is about also, that there is this ability to specialize in really narrow things, but you also have a broad knowledge of a lot of different things. So I feel fairly comfortable working on something from, structures of the brain all the way to what, a membrane molecule would look like.
So you work with basically the large aspect of medical.
Yeah, typically I work in the, if you remember, I don't know if you're here for the slide of the levels of biocomplexity, but I tend to be, system, organ, tissue, cellular, sometimes subcellular. I don't really get into molecular and atomic level.
Well, when you show it, you show it in like its systematic or somewhat somewhat interpretive,
Yeah.
realistic guy. You don't like do what that Ben Rayco did and take, or have you done it like that?
That dry data-driven stuff? No, I don't do anything data-driven. Like it's all macro instead of micro, for the most part. Except for the blood, that was like pseudo data-driven.
Right.
But you know, I don't typically. I'm more interested in growth structures and macro structures and the teaching of that than I am about getting into the visualization and the manipulation of 3D material. Like I may access it, I may use it for research, like the Osiris I was doing with the arteries, but notice I'm still painting them. I'm not like grabbing it and cleaning it up and using it.
Now, if one last question I put here, I understand that there's some things that are like impossible to necessarily visualize. Like you have to put a certain element of artistic interpretation on how such a thing works, looks, activates, interacts. Could you name a specific example and like what you personally believe you had to do to overcome that obstacle?
Yeah, well, I mean, I have almost everything involves aspects of that. It's just whether that is the predominant problem I'm trying to solve or not. Like you could think about were you here when I showed like the alcohol chart?
No.
No.
I'm sorry. Yeah, I know the switch actually probably shows some.
Yeah, so like where I'm talking about like the where I'm starting to link things conceptually to tell a story. That may be an example. The piece that I just did a sketch for, which was like a shoulder dystocia birth piece, like there's no way to you know, I'm showing nerves and bones that you can't see. So I'm always dealing with that.
That's exactly what I was asking about.
And that's the kind of stuff that almost every project I ever work on, at some point I throw my pencil down and start walking around the room because I can't figure it out. Like that's just part of what it is. You know, and I think about it and think about it. Sometimes I'll make a little neat eraser or whatever, you know, where I'm just dealing with a very difficult visual problem or difficult story.
Thank you.
So you can grow a molecule. I mean,
yes,
a molecule can be grown inside of a quartz crystal.
I'm not sure what kind of crystal it is. Right, and then they shoot X-rays at it, and but there are limitations to it. Like it does not pick up hydrogen. As I was reading, because it only has one electron. So for some reason, all the hydrogen is in X-ray crystallography. So there's programs now which are like kind of inserted them. Yeah, I don't know. Who thought of that?
Remedial electron microscope.
Like, how did they know what they were looking at was missing information?
I don't think the person brought a molecule in. Oh, maybe that's what you do. You take the molecule and you twist it around?
No, it has something to do with growing it. Everything that I read is going, but it's not an old technique. It's fairly new. I think the earliest reference I saw was 1971, but that would have been really rudimentary.
I haven't. That's exciting. There's way more going on in this world.
When you look at that animation, first animation, do you have a sense of how the whole fits together?
Yes. Like, I mean, basically I knew most of that information already, just by studying, scanning electron pictures. And because I'm a visualizer, I visualize it. But I don't know if other people could, but I think I had a sense of it. What I thought was really, really interesting is that I'm not an animator, I don't really think in motion. Like I think more in static landscapes. So that video is eye-opening in its function of movement for me. Because I just don't think that way.
Well, that's the part that made it, in a way, so hard to put together as a whole for me.
Yeah.
You said, this is how I learned about a cell, and then you saw all the components. That seems pretty clear.
Yeah.
But when you're in the middle, for me, when I'm in the middle of this universe where these things are pulling these things up, and the things are building, and then they're unraveling, I just wonder to what end?
Right, it's very fascinating. And this is going on all over. Every cell, more or less, those functions are happening.
No, it's
And
just,
where does it start and where does it end?
and what is it trying to do?
And how can you influence it? Can you influence it?
Eat better? Exercise?
Yeah, but that's such a macro thing. How does that translate to, atomic structure? I don't even know.
But again, your body of work thus far hasn't been concerned with so much those macro micro relationships, but rather just the systematic relationships between one like system with another.
Yes, like I'm more into patient education and explaining things that are not to the educational level as that. Like I'm more interested in how someone might learn about how can I tell them something that they'll suddenly absorb what I'm saying, rather than just say,
Well, it's interesting to think about how all sorts of forces act on cells. Even things like the way you think.
right.
And like attitude.
Right, that was a healthy cell.
Oh, God.
And we can even talk about disease and what happens. There's disease as a whole, symptoms that we feel, then, there's organs and tissues and cells and subcellular. And how does disease change those functions? And how do things like medications interact to alter that course?
Well, I mean, that seemed to be almost like a sum test there.
Right.
Because you're just looking at all the things that could go wrong.
I know, it's amazing, isn't it?
Interactions.
Yes, it's amazing. But there's a lot of redundancy, you've got a lot of cells. If one cell goes bad, you got lots of others. But if, a whole organ goes bad, well, then you're in trouble.
Oh, my God. That's amazing.
Cells are regenerating, they're dying, and then new ones are coming up. And that didn't even get into genetics and replication of DNA. And
You think the longer one got into all of that?
I don't know. I think that they were developing this as a teaching tool within their classes. So I would think I saw a new video from mitochondria, which is just mitochondria are those ones that are moving. That's the power center of a cell. Like that's where ATP is working. And so I started doing animations. David Blinsky, though, ended up putting a lot of the costs of developing the animation. Like he knew he was going to take a gigantic loss on it because what they wanted was so big. But no, it's career changing for him, so he did it with
It was an investment.
it was.
It's just amazing.
I don't plan on getting a PhD in now.
I was wondering about it.
Yeah.
Is there any material or website or collection in the gallery, we can view any of your existing works in
Mine?
for reference.
Hey, go to the website.
Your website? And
Yeah, I
that
have a website. We can go there.
and that is?
Permission.
Martens dash Kiefer, K-I-E-F-E-R, it's partnership with Amazon.com.
Right.
Cool.
His work is also medical related, but different. You know, they're just different people, different work. You know, he tends to be way more conceptual about stuff and way more editorial.
It'll be interesting to draw a comparison between the
Yeah, you know, we worked together for so long that our work was very similar when we started, and then I started teaching, and he started working alone, and all of a sudden, wow!